venetoclax is another very promising agent, approved already for other hematologic malignancies, but also finding its way into myeloma treatment. It is, in short, a new drug for the reason of mechanism of action, so not similar to any prior previously approved drugs. And this drug is specifically working through BCL2 inhibition, which is an important component of apoptotic pathway for self-destruction of cells, which we try to stimulate in malignant cells. We have found that this drug can be active on its own in myeloma in over 20% of patients. And what I think appears to be most exciting about the promise of the drug in combination with some other classes of drugs, particularly with proteasome inhibitors. There have been quite promising results presented and published in part for this combination with bortezomib, and now we have not published but presented at meetings and will be updated at ASH in a couple of weeks. Results of combination of venetoclax with carfilzomib. The activity of this combination is, I think, exceeding our expectations to what we would have expected that these patients would have achieved with using only carfilzomib and dexamethasone. While this needs to be still validated and proven, the drug appears to be enhancing activity of proteasome inhibitors.
One of the interesting areas is that as we've made these advances in myeloma therapy, we haven't really focused on precision medicine in myeloma. And the role of venetoclax, particularly in the 11-14 subset of myeloma, represents the first attempt at precision medicine in myeloma. And so what we know about venetoclax is that the responses are enriched in 11-14 myeloma. About 40 to 50% single agent activity, jumps up to 65% activity in combination with dexamethasone, and that represents a huge step forward for us in the context of using a genetic marker, the 11-14 translocation, and trying to pick a therapy based on it.
There's a lot of other newer drugs which have made tremendous difference in some other hematological malignancies, like BTK inhibitors like ibrutinib. That drug has been slowly taking into the myeloma field, although the data is not necessarily giving as much promise in terms of how well this drug will be working in myeloma as many of us have hoped. I wouldn't discount that drug completely out of the radar, but I think that the jury is out whether this will be a valid addition to our armamentarium and treatment of myeloma.
Other precision medicine approaches that are being tested in myeloma right now are the use of MDM2 inhibitors, particularly in patients with 17p deletion. That's a trial that's being run through the MMRC right now, where it's the MDM2 inhibitor alone and then in combination with ixazomib, with the intent of trying to overexpress normal p53 in patients who either have p53 deletion or p53 mutation. This, I think, does represent an important clinical trial idea because we know that loss of p53 occurs more frequently in relapsed and refractory patients, and so strategies to overcome this really are very important. Other molecular approaches that will be tested in a larger trial through the MMRC called MyDrug include mutations such as BRAF, mutations such as IDH1 and IDH2, as well as the NRAS and KRAS mutations by targeting MEK. And so these are all in development right now. There's anecdotal data here and there about targeting BRAF with a BRAF inhibitor, similar to what's done in melanoma or in colorectal cancer, but we don't have a large series of patients that have been treated in a trial, and hopefully MyDrug will be able to give us that.